Welcome all. Today we will explain about steroid biotransformation. Biotransformation is defined as the modification of organic compound into a recoverable product by chemical reaction catalyzed by the enzyme present in the cell. Firstly, we have to know that what are steroids and what is biotransformation. So let's start with steroid. Steroid is a type of hormone. Hormones are the chemical substances released by any part or gland of the living organism and transport through the blood and is responsible for the metabolism. Hormones are of three types: peptide hormones, lipid and phospholipid derived hormones, and monoamines. Steroids are from the second type, that is lipid derived hormone. Steroids are the biologically active compounds. They are synthesized in the human body itself. Next, we can discuss the structure of the steroids. Steroid is characterized by 17 carbon atoms arranged in 4 rings. Here, the figure shows the basic structure of the steroid, and the 4 rings are marked by alphabetic letters A, B, C, and D. It contains the specific site for chemical modification by microorganism. All steroids have some basic structures, that is, one cyclopentane ring and 3 cyclohexane ring. In this figure, the cyclopentane ring is marked by the letter D and the cyclohexane rings are marked by A, B, and C. In this figure, you can see the 17 carbon atoms arranged in 4 rings. That is the basic structure. There are different types of steroids. First one, sterols, then bile salt or bile acids, mineralocorticoids, corticosteroids, and sex hormones. First, sterols. The most commonly known of this is cholesterol. Other sterols help the body to make vitamin D from sunlight and to build up cell wall. Second one, bile salt or bile acids. These are made in the liver and they do not function as hormone but are necessary for the digestion and absorption of fat. Third one, mineralocorticoids. This maintain the balance of sodium and potassium in the body. And the fourth one, corticosteroids. It includes cortisone and cortisol. It have an important role in the immune system. And finally, sex hormones. These are the male hormones including testosterone, which together are called androgens, and the female hormones including estradiol, a type of estrogen. Okay, so these are the types of steroids. Next, the functions of steroids. The best and most abundant steroid in our body is cholesterol. Cholesterol formed in brain tissue, nerve tissue, and bloodstream. Some steroids used in the treatment of disease. Example, cortisone and its derivatives used to treat rheumatoid arthritis. That means the steroids used for a wide range of therapeutic purposes. It utilized as immunosuppressive, anti-inflammatory, sedative, and contraceptive agents. Steroids used to treat some forms of cancer, HIV, etc. Naturally occurring steroids are the example Saccharomyces cerevisiae contain high level of ergosterols. Okay, so these are the functions of steroids. I think you got an idea about steroids. Then we have to know that what is biotransformation and the difference between chemical transformation and biotransformation. Transformation of steroid means conversion of precursor steroid to important drug intermediate and further conversion of this intermediate to the active compound by chemical or microbial process. Chemical transformation of steroid cause serious environmental and health problems. And the biotransformation, that is the transformation by microbial process, can overcome this problem. That means biotransformation is the conversion of precursor steroid to an active compound by microbial process. And also it can be defined by a process which produces the structural change in chemical compound to form a new compound. Biotransformation contains two words, that is first one bio and second one transformation. Bio means living thing and transformation means change in structure. So it is a process which produces a structural change in chemical compound to form a new compound. This conversion is mediated by microorganisms. Therefore, many new or novel compounds can be synthesized through biotransformation reaction. Transformation may be the addition, deletion, or modification of functional group at a specific site. The changes are chemically isomerisation, amination, decarboxylation, dehydration, condensation, oxidation, hydroxylation, or dehydrogenation. Here a table is given and the table shows the transamination reaction, the substrate, that is the precursor steroid used, and the newly formed product, and the microorganism involved in this reaction. Okay. Arthrobacter simplex can convert the precursor steroid, that is hydrocortisone, into prednisolone by one dehydrogenation reaction. Another example, Corynebacterium simplex can convert hydrocortisone to prednisolone through one dehydrogenation reaction. And the third example, Curvularia lunata can convert rich stein compound to prednisolone by 2-beta hydroxylation reaction. So these are some of the examples for the biotransformation. Next, we can go to the fermentation process. Steroid transformation fermentation are carried out in stainless steel fermenters. Media used in fermenter is based on which microorganism used in the biotransformation. There are different types of microorganisms involved in the production of steroid transformation. So, the media will change based on the microorganism used. Molasses are mainly used for carbon source. Steroid substrate are not soluble in the water, so it dissolved in water miscible propylene glycol. The solvent used should be non-toxic to the microorganism. Otherwise, it will affect the fermentation process. These steroid precursors are added only at the end of growth phase of the microorganism in the fermenter. Then only it undergo minor modification. There are different microorganisms are used for the fermentation process. Fungi and bacteria are mainly used. Fungi include filamentous fungi, Rhizopus, Curvularia, Aspergillus and Fusarium. And one of the example for the bacteria used for the fermentation is mycobacterial spores. Then the final topic, production or the biotransformation fermentation. It involves 4 steps: cultivation of microorganism, incorporation of steroid and inhibitors to the medium, then transformation, and finally extraction. First, the culture is added into the fermenter and is provided with nutrient media. Nutrient medium consists of glucose, sucrose and carbon source. Proper aeration and agitation is also provided for the growth of microorganism. Then the second step, incorporation of steroid and inhibitors to the medium. Steroids are dissolved in solvent and then added into the fermentation tank. The steroids are added after the growth of the microorganism. The desired inhibitors which inhibit the other enzyme producing microorganisms are also added to the medium. Okay, so that will promote the proper fermentation process. Then the third step, transformation of steroid. After the addition of steroid and inhibitors to the fermentation tank, the microbial enzyme which are secreted transform this steroid. So the transformation of steroid to a new product will occur. This process requires few hours to a day. After the transformation process, the product is extracted. That is the final step. For the extraction process, water immiscible solvents are used. Then it is followed by column chromatography. This is about steroid biotransformation. Thank you. Thank you all for watching this video.